Hi, I'm Esmeralda, and I'm doing my senior seminar project on dermatophytes on barn flies. Dermatophytes are parasitic fungus causing cutaneous infection, affecting only dead keratinized skin, or tissue, in the hair, skin, nails by producing keratinolytic enzymes. They are also found in soil, besides hair, skin, and nails. They are found in the division Deuteromycota in 3 genera: the Epidermophyton, the Microsporum, and the Trichophyton. Their usual transmission is via animals to animals, humans to humans, and animals to humans, and humans to animals. One type of vector that has not been yet studied as much is possible transmission via flies. And we know that flies are actual vectors for transmission of bacteria, protozoans, and some viruses. Flies have 3 mechanisms of transmission. One is through their legs. They have a sticky substance on their legs that allows them to adhere to the surface and also allows for stuff to adhere to them. The second form of transmission for flies is through the mouth parts. They actually, when they're ingesting their food, they can actually ingest microorganisms that sometimes need to go through the digestive tract of the fly in order to be able to reproduce somewhere else. So these flies sometimes vomit their food out, and in which these microorganisms come out, and they can either come out with the vomit, or they can come out, or they can stay in the mouth parts. And these mouth parts actually come into contact with another food surface, and they can get dropped off there. The last form of transmission is via their wings. The wings come into contact with various surfaces, including the air, and the air inhabits a lot of microorganisms as well as spores produced by fungus. And many fungus rely on wind dispersal for their spores to go to separate areas so that they can reproduce. The purpose of my experiment is to analyze flies and be able to isolate dermatophytes on the flies, whether it be on their legs, mouth parts, or their wings. The materials and budget for my project, I need a nutrient agar, potato dextrose agar, and the dermatophyte test medium. The PDA and the DTM is already in stock. The only thing I need is a nutrient agar, and it's well below the budget. The reason why I'm using nutrient agar and potato dextrose agar is because those are the common types of agars used to promote growth of dermatophytes. And obviously the agar preparation tools and autoclave plates, as well as plates, sterilized utensils, and gloves are needed. But all of that is in stock. So my methods and timeline, I'm actually already started collecting barn flies in December, and it's going all the way through February and March. I'm going to extend it up to March, only if I do not find any dermatophytes in the first collections. However, I'm actually plating my agars starting January. I'm going to start off with the nutrient agar. There's no specific reason for this. Both agars are used to promote the growth of fungus. So it takes about 2 weeks for dermatophytes to grow. So after these 2 weeks, I'm going to actually be analyzing the plates based off the colony topography, the texture, and the color of the colony to determine if dermatophytes are present. One known colony topography is the Microsporum canis. It produces white, low, fluffy colonies with white on top and then yellow and orange spots on the bottom, on the reverse side. Trichophyton produces a flat, powdery, off-white colony. So that's what I'm going to be looking for in my plates. And that's the macroscopic identification. Another identification I'm going to be using is microscopic, and that includes the type of macroconidia and hyphae that are present in the colonies. And Microsporum produces a rough, thick-walled macroconidia with terminal knobs, and the Trichophyton actually produces smooth and thin-walled cylindrical macroconidia, so I'll be able to compare the two. The difference between the Trichophyton and the Microsporum, Trichophyton actually produces clovate microconidia, but they also produce club-shaped macroconidia, which is 2 to 9 cells. The purpose of this experiment is to verify if flies can actually be vectors for dermatophilic infections, and how the medical community can also use this as prevention, especially for immunosuppressant individuals that are more capable of getting these diseases because their immune systems aren't as functional as normal individuals. And the way that other infections can occur is by the dermatophytes actually cause irritation on the skin, and the person actually scratches and allows an entry wound for other infections like bacteria and other fungus. And this is really important so that we can find prevention measures and also new ways of finding cures for dermatophytes.